current cell-free process and how it performs in downstream mRNA applications, followed by the solutions for mRNA and the lipid nanoparticle encapsulation process development and manufacturing capability. And finally, I'll talk about our service, our integrated cell-free from plasmid to mRNA platform. So to start with, I want to share with you the GenScript ProBio's product portfolio in the cell and gene therapy field. It covers the cell therapy, gene therapy, and the nucleic acid, such as mRNA and DNA drugs. So for cell therapy, we have ProBio have the lenti-helper and the lenti-bone plasmid to support the lentiviral packaging system with a high-yielding E. coli strain and with also clear traceability to support the cell therapy plasmid manufacturing. We have also our proprietary Power S293T HEC adherent and suspension cell lines, which have successfully delivered more than 100 batches of GMP production. In terms of gene therapy, ProBio has the A-assistive plasmid to support the AAV packaging system. We have a Power S ITR plasmid production strain focusing on the solving the problem of plasmid with, for instance, with a recombinant issue and stability issue. So this strain is very useful and performs very well on plasmid with certain structures. For instance, on AAV transfer plasmid with ITR structure, or with mRNA plasmid with poly A structure. So, and the proprietary Power S 293 suspension cell line that can produce AAV with a titer greater than 1 to the power of 14 viral genome per liter. And we have supported multiple batches scaling up from 2 liter to 200 liter AAV production. In the field of nucleic acid drugs, we have a plasmid ProPlus process that is specifically targeting mRNA as plasmid starting raw material. And then there is a focus of our today's topic. We will introduce our ProSyn cell-free manufacturing process from DNA to mRNA. And for the mRNA, we have a complete solution from the in vitro transcription, the IVT process, to LNP encapsulation to support the development and production of personalized mRNA vaccines. So personalized cancer vaccine, PCV, is a type of personalized treatment. Different companies may have a different name for it. For example, there are companies call it INT, the individualized neoantigen therapy. And there are also more like these kind of different names, but they all mean basically one thing, this personalized cancer vaccine. The general workflow involves, for instance, taking the blood or resected tissue from the patient and conducting the sequencing and AI comparison analysis to identify the sequence of the neoantigens that can trigger the T-cell response, which are encoded, these neoantigen, which will be encoded by the mRNA and then infused back into the patient's body. And this neoantigen sequence is unique to each patient, so it is a very personalized treatment. What it brings, what kind of challenge it brings to us is the speed and the production efficiency, because often the maneuver starting from the sample collection all the way to the injection, so the needle to needle timeline will be within weeks. So that's something very specific to these personalized vaccines. So this slide shows what is the advancement of the personalized cancer vaccines globally. As you probably already know that there are several pipelines that are in clinical, late clinical phase, Moderna and BioNTech. So this Moderna 4157 is in clinical phase 3, targeting for more than 1,000 patients. It is used in combination with the Keytruda PD-1. It usually encodes 34 neoantigens in series. The needle-to-needle reported timeline is about 42 days. For the BNT122, that is currently in phase 2 and is used in conjugation with Tecentriq, the PD-L1 antibody drug. As you can see from this picture, from tissue surgery to the first dosage of the mRNA, it takes about 9 weeks. During this period, sampling, the detection of the sampling and the sequencing, gene synthesis, all the way to the production of mRNA, DS, and DP. So drug substance and drug products will be completed, or they have to be completed within this short time frame. So after talking about the personalized cancer vaccine, I want to give you an introduction to to compare the traditional E. coli-based DNA synthesis and our cell-free system. In this page, you can see that the E. coli is traditionally used to produce the plasmid. It's a natural vehicle for the plasmid production. And during the plasmid replication process, there are several mechanisms for the E. coli to ensure the accuracy of the plasmid replication. First, the base selection, which contribute to 10 to the power of -4 accuracy of the system, followed by the 3 to 5 prime exonuclease to cut down the wrong sequence, and then finally there's a mismatch repair mechanism. So in total, these three processes guarantees the replication accuracy fidelity, which adds up about 10 to the power of -10 frequency for one error rate. And compare with the traditional E. coli system, there is also so-called the rolling circle amplification system that we used in our cell-free process. So this process uses the 529 DNA polymerase to conduct what we commonly call a rolling circle amplification. It is worth mentioning that the 529 DNA polymerase has the same base selection and the 3 to 5 prime exonuclease activity, but it lacks the mismatch repair mechanism. So the fidelity for this cell-free synthesis application process is around 10 to the power of -7 accuracy. So the structure of the 529 DNA polymerase determines that it has excellent strand displacement activity and excellent processivity for the DNA replication. At the same time, the proofreading of the 3 to 5 prime also ensures the accuracy of the replication process. So compared with the other DNA polymerase system, this combined functional enzyme brings excellent high accuracy and stability to this for the industrial application and amplification of the DNA. So that's the starting point of us for us to use to build our process. So we have established this ProSyn DNA manufacturing platform using the 529 DNA polymerase. So based on this system, we built this platform starting from a very small amount of traditional circular plasmid, and we use random hexamers as a primer to initiate the linear DNA replication by this rolling circle amplification mechanism. And after the enzymatic digestion, the DNA will be linearized and be cut. So the resulted linearized mRNA will be is identical to what is traditionally produced by the E. coli system, which is follow the similar process. So first produce the circular DNA and then be linearized by the enzyme. And we have further purification processes to remove the truncated small DNA piece, making sure that all the resulted DNA, linearized DNA, are the accurate and the full integrate with the full integrity of DNA. So the traditional plasmid to mRNA production process is listed here. So plasmid will have to go through the traditional cell banking, high density fermentation, harvest, lysis, purification, being released to enter the linearized DNA and going through this purification step, and then as a starting point for the IVT mRNA. With the introduction of the ProSim platform, so this workflow can be greatly simplified with the rolling circle amplification and enzymatic digestion followed by purification. The resulted DNA can directly be used for the IVT process. This eliminates the traditional cell banking, clone selection, and stability step, which that greatly reduces the early step work that we have to do. Therefore, the whole workflow from the template to the mRNA LNP manufacturing can be completed within several weeks, within actually 2 weeks. Okay, so next few slides, I will talk about the advantages and and we'll share you some case studies for the using this process. The challenges and considerations, including the advantages of using the cell-free process, they are presented in these slides. So first of all, the fidelity and quality control standards of using this new process is a key to consider. And for this item, a lot of research have been done, like I showed in previous slides, to demonstrate the accuracy of this 529 enzyme. It is considered currently it is considered as a highly accurate process, and compared to, for example, the traditional PCR and other replication process. And we are still we are also in the way to continuously collecting the data and compare it to the traditional E. coli production system to demonstrate the accuracy of it. Also, the downstream application we have done, I will show you in the next few slides, in the mRNA downstream applications and the sequencing data to confirm that the quality attributes of the mRNA remains the same or even better compared to the E. coli-based production system for DNA. In addition, it waives the process-related impurities generated by using E. coli, for instance, the endotoxin, the whole cell DNA, whole cell protein, and whole cell RNA. And due to the elimination of cell-based biological variation, this process gives better stability for the product we produced. So there's a more clear system, a clearer structure to activity relationship. Of course, for the enzymatic kinetics, the control is easier than the E. coli, like the biological production system. Therefore, it improves the reliability and quality of the product, and also it simplifies our process failure handling and changeover, because it doesn't have any E. coli or living bacteria. So for the processivity given by characteristic from this specific enzyme, it gives excellent robust scale-up robustness and very high processivity up to 70 kilo base pair DNA synthesis. Very efficient, and it also gives a higher degree for future automation possibility. to simplify the process and to automate this process. And due to this due to the process simplicity, it also reduces the future scale-up risk and the tech transfer risk. This accurate process also get rid of the E. coli based, for instance, some recombination sensitive sequence issue, because in E. coli there are a lot of recombination related enzymes that can be troublesome for the ITR and poly-A structures. So by using the cell-free system, the stability of the plasmid can be greatly alleviated. It also eliminates the antibiotics usage during the production. Of course, we have to consider that we introduce a new enzyme 529 that has to be monitored throughout the culture, throughout the process. Last but not the least is that FDA has already proved one IND program using enzymatic DNA manufacturing in the early last year. So it's a good indication of the regulatory acceptance for this new technology. We have done quite comprehensive comparisons between the DNA produced using a traditional E. coli system as well with the cell-free system. For instance, the sequence accuracy here we showed is identical result using either the NGS sequencing or Sanger sequencing. We will also keep monitoring the data to collect as much as possible across different projects to demonstrate the comparability between the cell-free system and the E. coli system. And the cell-free process also is demonstrated to be a very robust process, given the fact that the scaling up is very straightforward. As you can see here, the scale from 10 to 100 milliliter, the final yield, amplification rate, and the plasmid and its plasmid quality generated from this process is very consistent. Further on, the DNA produced with the purity tested by ion exchange and the CGE gel electrophoresis they show very good very high purity compared to the traditional E. coli system. We further did the comparison at the mRNA stage. So here this data shows that the mRNA produced using these two different systems, DNA, the mRNA integrity, purity, the capping efficiency of the mRNA, and poly-A length of the mRNA are all comparable between these two DNA raw materials. Also, we use the mRNA to be encapsulated in the LNP and did in vitro expression assay. It shows that the bioactivity of the mRNA generated from these two processes are also very comparable. So, and in the next section, I'll show you some data on our mRNA and LNP process development and manufacturing platforms. There are some. We have here. So in this section, we have in-house ongoing actually quite some linear mRNA projects. And saRNA self-amplifying RNA project, as well as circular RNA projects. As you can see here, that through the accumulation of our in-house R&D data and customer projects experience, we have built a comprehensive platform for our IVT mRNA process. For the traditional mRNA, we can use co-transcriptional capping and enzymatic capping. the purity, capping efficiency, and the productivity and the recovery are all very good. The drug substance or the mRNA concentration we get as high as 5 milligram per milliliter. And the self-amplifying RNA is iconic for its long mRNA length, up to 12 kilonucleotide length. So for this one, we have faced the challenge to get to purify the mRNA to get rid of the truncated or short length mRNA. So that's the challenge for the self-amplifying RNA modality. And even for this, through the downstream optimization, we have achieved a high purity mRNA and higher than 95% of mRNA integrity, with also uncompromised mRNA capping efficiency and yield. For circular RNA, we can do we have different circularization process like intron self-splicing or T4 RNA ligase method. For both of them, we have data to achieve a purity higher than 90% analyzed by PA 800 equipment. And we have also in the process of scaling up the purification process for the circular RNA. So far, we have demonstrated in gram level circular RNA production. The production process is very efficient. For the LNP process, here is a flow chart. I want to show you what are the considerations during the process development on our platform. I won't go into the very details of each items here, but I think in the end, the point is if we want to achieve good quality results on the lipid nanoparticle size distribution, encapsulation efficiency, and consistent lipid nanoparticle nanostructure and morphology. There are many parameters in each step that are intercorrelated. We need to study them. We have to have a comprehensive study during the process development and take into account the scalability of the process. Here are some additional development stage data behind the robust scale-up productions that we have done. We have spent huge efforts on correlating each parameters with the product quality attributes, as you know, through the quality by design approach. So here, by screening, for instance, the N to P ratio on different mRNA, two different mRNA molecules, we can have a correlation between the NP ratio to its size and the PDI distribution. And during the process development, we also tested different total flow rate, the encapsulation total flow rate and flow rate ratio screening, and try to correlate it with the performance of the final product. We also further on tested different buffer conditions during the encapsulation, the TFF and final formulation buffers, to understand the impact of LNP size, PDI, encapsulation efficiency, and stability of the product. So this information across different projects, we are accumulating those data on different modalities, which in the end, this will be platform data to help us to better understand our LNP process to accelerate the development for every incoming new projects. Sorry, take a sip. So the platform data are summarized here on different LNP formulations. So here we show that we have the traditional, some traditional commercially available formulation like BioNTech and Moderna's formulation. And in addition, we have also customers' proprietary LNP formulations and ionizable lipids. These are all shown very good results on our LNP platform, which demonstrates that our LNP platform is quite universally applicable, meaning that it can support the first establishment and continuous manufacturing for the mRNA project, especially for the PCV program, where the timeline is very important. With this platform, a new modality comes in, we can quickly establish the process and then proceed into the GMP manufacturing stage. So here is in the next few slides, there are some real world production cases or scenarios that I want to just show you. So this is our GMP facility with the isolator automated fill and finish system. We have completed more than 10 batches of gram level GMP scale up, all the way from encapsulation to fill and finish. With the DP concentration achieved high as 2 milligram per milliliter. The system is running very well, and you can see here that there's one batch with 500 milligram production. The size distribution is very consistent, and the yield and the encapsulation efficiency performance are very good. There are some more real-world data to demonstrate our quality attributes and the performance of each batch. So what I included here are 3 different projects. So you can see that some project-related data are not. So they are not from the same project. There are 3 different batches for 3 different projects. As you can see that the particle diameter, osmolality, integrity are all very good, and we are keep accumulating this GMP production data to support a quicker and more robust scaling up. There are some more data on the stability of the products that we produced. So this slide shows the mRNA drug substance stability. We have done stability for the drug substance in -70 for over a year. So we are keep accumulating the data, but the data shows so far the stability of the mRNA is very good. 3-cycle freeze-thaw, 25 Celsius degrees, and shipping stability. They're all quite. They can support the commercialization of this product. For the lipid nanoparticle drug product stability, we also have more than 10 projects data that are being captured and collected in parallel. So here I just demonstrated 2 modalities in which one is 0.1 milligram per milliliter, the other is 1 milligram per milliliter in DP concentration. As you can see that so the first of all, you can see the size is quite consistent for these 2 different modalities in 4 Celsius degree, plus minus within plus and minus 5 nanometer in size within 3 months. And 5 cycles freeze-thaw for these 2 are also very good. It shows a very slight increasement of the size over the freeze-thaw cycle, but overall it is still controllable within plus minus 5 nanometer increasements. And the PDI are all quite comparable, considering that the variation of the analytics, so it's below 0.2. In terms of long stability, we have data accumulated up to 12 months, but due to the time why we only put the data up to 6 months here, but actually the 12 months data are also available, which shows still very stable LNP product. The light exposure we also did for these two products. Overall, the stability for mRNA as drug substance or being encapsulated in LNP, the stability data on our platform are very good. Supports the further commercialization of the product. So this is a summary page, which I want to highlight some points. So for mRNA LNP platform, we have excellent formulation development platform that we have tried multiple payloads, not limited to the mRNA, self-amplifying RNA, circular RNA, but also we have done DNA encapsulation and sgRNA. Also, multi-component LNP encapsulation. We have quite some projects ongoing that achieved very good quality control data. So for process development and scale up, we have successfully completed over 10 GMP batches. The DP concentration can be between 0.1 to 2 milligram per milliliter. We have a scale up capability up to 10 gram per batch. The process during the process, the lipid nanoparticle size can be controlled very tightly, plus minus 10 nanometer across different scales. We have also demonstrated quite a good DS and DP stability. For good process development, we also need a very good analytics platform to support. As you can see here, we have all in-house capability to do the analytics, including the physiological and morphological testings. Not only that, but also we have in vitro and in vivo pharmacology group that can help us to troubleshoot any process deviations that we might see during the process development stage. So below there are some figures, just go through them. So this is the empty ratio, the LNP empty ratio figure. So the empty ratio is about 3 in one of our GMP production runs. So nearly 97% of the LNP are full. So this is the cryo TAM on microscope picture that we use that frequently during our process development stage to look really into the morphology of the LNP, making sure that all the parameters, all the process that we in the end determined or locked, they generate LNP in a homogeneous fashion. So we have in vitro and in vivo functionality group that can help us to also to troubleshoot if there's some process issues happen. So in the very last section, I will briefly talk about our solution and our service all the way from the cell-free DNA to the mRNA in the field of personalized cancer vaccine. So first of all, we support our customer in a drug development life cycle, all the way from the IND enabling study towards the clinical phase production and to the late phase clinical production and commercialization. And for both plasmid and mRNA, for each respective life cycle stage, we have specific product grades to support the customer's project in the phase appropriate manner. We have like established this ProSyn, which is very compatible with our mRNA platform in terms of supporting the PCV program. All the way from the ProSyn DNA synthesis to IVT and LNP mRNA, we can complete within around 50 days from the, like for instance, from the genes, obtain the sequence from our customer to the final DP release. So this is another way to show that our platform, we aim to get a pure plasmid manufacturing through the ProSync platform, and with the PCV mRNA, we have very robust platform to quickly establish the process and to do the process analytics and the drug product release. With the feeling of cell-free DNA and our robust mRNA LNP, I think in 2024, we are able to support some very intensive personalized cancer vaccine timelines by integrated cell-free process to shortcut some inter-process control and QC, to do the drug product stability by demand, or in the future, we are also considering for instance, a fully automated miniaturized GMP manufacturing for these application. We are trying to establish our in-house material and equipment to support the automated manufacturing in the future. And we also are considering the near site production, like near hospital production for these specific modality, and try to get a more flexible and small feet GMP facility to further accelerate the mRNA drug product release for these personalized drug medicines. With that, I want to thank you for your attention. If you have any questions, I'm happy to take.
Okay, thank you for Dr. Pan's presentation. Is there any questions? Okay, not yet. Thanks for Dr. Pan's time.
Thank you.
Thank you. Bye.